Today we will discuss about this glycan can binds on the RNA and then help them to express on the cell surface. So previously what most of the people know or what the science know is that glycans can bind either to the glycoprotein or to the lipid and then helps them to express on the cell surface. However, recently some papers has published that RNA can also bind to the glycan, and this glycan can help RNA to express on the cell surface. So this is one of the new finding that has been done in the glycan biology recently. However, it is not yet completely accepted by everyone. So let us now discuss what is the experimental procedure that they have used and what are the evidence they used to show that RNA can express on the cell surface with the help of glycan. So they grow cells in the cell culture medium or the tissue culture in the medium, and they have also done their experiment on the live mice or animal model. So what they let us discuss the experimental procedure for the considering only the cell culture cell culture growth experiment. So what they do it, they grow the cells on the normal cell culture medium, and then they add this acetylated NAZ. So this can only bind to the, so this is developed with the help of click chemistry, such a way that it can bind to the sialic acid site of the glycan. Then they did is that they add the triazole and did the column cleanup, and then in order to get rid of any proteins, they treat the sample with the proteinases key at 37 degree, and then did the again column cleanup so that all the samples get free of all the proteins, and then they denature it and run it on the gel and measure the sample using uh, using different methods that we will discuss as we go through the uh, figure. So this is the general experimental procedure. So and this one is the we can say mRNA blotting. So in this, what they have did is whatever the samples that they get, then they run it on the mRNA blotting and check whether there is any presence of this uh, glyco RNA. So they have said that RNA that is bind to the glycan as glyco RNA. So that they have measured using this RNA blotting. So, and this is the samples that are collected at different time period. We can see that during the initial time period there is no presence of glyco RNA. After that, as the time increases, there is an increase in the amount of glyco RNA. And for the whole experiment, they have used a concentration of this is acetylated and NAS as around 100 micromolar, and they can see the presence of this glyco RNA. So, and in order to check whether they have injected the same amount of protein, they did the staining and measured it using this CBR gel, CBR stain. So we can see that there is the equal amount of RNA that is present within the sample. And later on, what they did is to confirm that this is because of the RNA. So what they did is they test the glyco RNA, and the samples, they treat these samples with the DNAs to check whether this is these signals are because of DNA or of something else. So, and also with RNAs. So when they did with the DNAs, there is no change in the glycoRNA. So with this, they come to a point that it is these signals are not because of DNAs. However, when they treat it with this RNA cocktail, they can see there is no glycoRNA. So this substantiate their point that this signals is because of RNA, and that is bind to this AC for acetylated man enzyme. And next, what they did is they block this RNase cocktail by treating the sample with super resin and to check whether or to and to further substantiate their point that the signals are because of RNase. So when they block this RNase through this super resin, they can again see the signal for glycoRNA. So this made a point, this substantiate their point that the signals are because of this glycoRNA bind to the this RNA bind to the glycan. And when they did the in vivo experiment on in the mice, and they have collected the sample from liver and spleen, and they can see the same signal, same kind of signal in the in vivo experiment also, that is the presence of glycoRNA with the increase in the time. And when they treated the sample with RNase cocktail, there is no signal. And this happened for both the liver and spleen sample. So from this they made a point that this presence of this glycoRNA is not because of some kind of uh, some kind of some additional chemical reaction going on in the tissue culture. However, it is this phenomena exist in the animal model also, or this phenomena exist in the in vivo RNA source also. And these signals are based on this acetylated mannose that is biotinylated with the visible signal. So in this figure, they made a point that glycoRNA, they made a point that RNA bind to the glycan, and from the chemistry that they have used, uh, further they have made the point that this RNA, this glycan that is bind to the RNA, are mainly rich in the sialic acid and fucose, that we will discuss in the next videos or next video. So that's all for this video. If you need to understand this whole experiment or whole story of this glycan binds on the RNA, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time. And one thing, that before we ended the video, I would like to let you know that this signal is considered as the non-specific signal, as it is represented by the star. And the glycoRNA finds it difficult to travel to gel. So one thing that they made a point that this could be, first they assume that this could be because of high molecular weight RNA, and later on they find out that this might also be because of the glycan that is bind to the RNA that prevents the flow of RNA through this gel. We will discuss all these things in the next video. That's all for this video. Thank you for your time.